name is Victoria English. I'm an editor and co-founder of a company, Evernow Publishing. Our magazine, MedNows, discusses the business of medical innovation, and we've been in business for 20 years.
So I'm Justin Wilson. So I'm a European and UK patent attorney at Withers and Rogers. I sort of got a background in biotech and have been working with lots of companies in the biotech area, including the gene and cell therapy areas for the last 20 years or so, including in some of these RNA areas as well.
I've already introduced myself, so.
Likewise.
Yep.
Hello, I'm John Moore. I'm a basic scientist. Been working in the industry about 25 years. Currently co-founder and CSO of Epitopia, which is a cancer vaccine company using native mRNA encoded in LNPs, focused on antigens from the dark genome. I'm a non-executive director of one of Peter Cullis's companies, Outfox Bio, based in Oxford, and an operating partner at Advent Life Sciences.
Good. So I'm very thankful to Kathy for the presentation, but I noted that mRNA therapeutics didn't appear on the slide. You did mention vaccines. Yeah, that's what I mean. That's what I mean. It's an invitation. Kathy's presentation is an invitation for us to explore this new modality, and it's so new that it doesn't appear in most people's slides, basically. Tony has used the word disruptive to describe this session, and I think that that's illustrative by the presentation from AstraZeneca and also what the remarks that Rose said. And I was wondering if we could start with Rose's observation that this is a new world for drug discovery in the sense that we're focusing on the cell. And I was wondering if you had any further comments to make on that, whether I've understood your remark correctly to start with. And if so, tell us a little bit more about your insight.
Yep, can do. So I think, cell therapy it's sort of new relative to antibodies, but isn't new. We've got 7 on-market products in DNA CAR-T. And mRNA itself is also not new, as my sort of slide illustrate with the different RNA therapeutics and the vaccines. I think what is new is now applying that to cell therapy, and that's where the innovation now is, and that is all at the early stage, so preclinical, clinical stage. I think then the challenges of that is that because we are reusing technology from different areas, all of the sort of the baggage comes from there as well. So, for example, if we're using mRNA LNPs from the vaccine world, all of that LNP IP becomes relevant, all of the technical challenges from LNP gets applied as well. So I think it's a new thing to think about with regards to the complexity of these therapeutics. So we haven't just got like one molecule, you have all of the different components within what we have, not just the mRNA, but all the other stuff, and that's where the new approach maybe has to come as well.
So if we can just continue a little bit on this subject, looking at the platform technologies and looking at patenting platforms. You gave us as a very good example of a modern approach, Orna, recently acquired by Lilly. Somebody has been thinking about this. Maybe you could give us some more information about why you think this patent application was successful and why it is an interesting approach, perhaps, for other companies to follow.
Yeah, I can do. So I think the approach of patenting your platform itself isn't new. I think what's interesting is that the attention the pharma companies are paying to that approach. So previously, a biotech might build out their platform, then have to have a proof of concept actual product, and so then you'd have to divide your strategy up between here's the platform that can be licensed out, and here's a product that maybe we want to get licensed separately to a pharma company. What is interesting in this space, as I tried to highlight, was the pharma companies are coming in early and buying that platform. And I think the reason for this is just because it is so competitive. Everybody wants to be the one with the platform that's going to work. Mm-hmm. And we're at that early scientific stage of no one knows which one it's going to be yet. So
doesn't know, yeah. George, do you have any comments on this?
Yeah, I completely agree with that. I guess traditionally you would see big pharma buying SMEs up when they've got like phase 2, phase 3 ready assets, ready to go to market. reducing the risk to the big pharma. Yeah. You're absolutely right, Rose. The speed of development of these platforms means that big pharma has to sit up and take notice and get a toe in the door before someone else. There's a bit of a frenzy going on at the moment, I think, basically, around these platforms. We'll see whether it's real or whether it's just a bubble that will burst at some stage in the future. But I think for some of them, it will be a hit on goal. They'll do really well. Some maybe not so much. I think mRNA in particular has shown utility. I think with regards to the RNA situation, circular RNA still haven't shown utility in the clinic.
That's what I've heard.
In the clinic yet. So it's a big punt,
Yeah.
but it may pay off.
Well,
Yeah, I think interesting with the Orna though, I think was, yes, they have that foundational circular RNA, but I think why they were successful is that they've got all this other IP as well. They haven't just focused on the circular RNA part. They've got their ionizable lipids, they've got their LNPs, and that may be what makes them attractive as well.
Justin, do you have a view on the positioning of the IP and how it's...
Yes, I mean, certainly for some, my experience. I've been working with RNA patents since the late 2000s. I mean, and at that stage, it was very much sort of universities developing their technology and developing these sort of platform technologies relating to, the particular mRNA features of caps and UTRs and poly-A tails and things like that, in the early stage of the LNPs. And then, I guess since then we've seen the progression which was, I guess, accelerated after the mRNA, the success of the mRNA COVID vaccines, to now where it's much more, small companies who are filing the patents rather than, or presumably in-licensing some of that university IP, but they're the ones now filing the patents and developing that, and taking that more commercial approach where hopefully they've got the platform technologies, but then they're also developing specific products which they're trying to protect the individual components of those themselves with new filings to, as Rose says, build up their overall patent portfolio. And I guess one thing as well in terms of the investment coming into the space, that's certainly in big pharma. There's been a bit of a patent cliff where some of their blockbuster drugs are coming off patent now and over the next sort of 5 and 10 years. So I think certainly there's been some increased interest in trying to bring in some new assets into those big pharma companies. To try and fill some of that pipeline.
John, would you like to tell us about?
Without Fox hats on, our view is that a lot of development is possible in formulation rather than just lipids in particular. And so returning to some of the old ones from Onpattro, been validated in hundreds of patients, can give you very surprise performance. An issue with existing formulations is the PEG lipid, which does generate antibodies which build up with time. And the pedicolysis companies, Nanovation and Outfox, have a way around that. Platforms are very important. From an Epitopia point of view, we've seen a lot of IP. Some of the most foundational stuff, like the CureVac ones from 20 years ago, is now expired. And that gave a big boost from what was possible with natural things. And our LNP provider, we went for the one with the dominant position. And it's a very expensive technology to acquire in terms of future shares of revenue. And therefore, I think there's a lot of interest in other solutions. Yeah.
I just want to move on here to the definition and scope of the therapeutics that are possible here. I was actually surprised when I did some research on this to find out that there were quite a few, 31 clinical trials underway in different areas of RNA therapeutics. I was surprised. And I was wondering if we could discuss for a minute amongst the different applications. You've got the antibodies, you've got the cell therapies, you've got the replacement therapies, enzyme replacement therapies, you've got solid tumors. AstraZeneca is looking at a lot. How do you decide? I suppose it's based on your existing portfolio and how you can augment your existing portfolio with the new technology. Maybe you could give us a little insight into how you deal with this.
Yeah, so obviously we're trying to use RNA or other similar platforms to augment, as you said, our existing therapeutic areas and portfolio within those therapeutic areas. So I guess that's, AstraZeneca is divided into oncology and biopharm. So on the oncology side, we definitely as been pointed out in one of the slides, we're definitely working with the CAR-T platform, the CAR-T, and now exploring in vivo CAR-T with mRNA in a number of different projects. And basically, I think big pharma like AstraZeneca have to do this to keep up with what's going on around the world, particularly what's happening in China, to keep up with what's happening there and how fast these therapies, these new therapies, new modalities, are being brought to patients in China with IIT trials. And so we can't, I guess we can't sit back and just kind of watch this space. We have to be players, we have to be part of it. And we have to augment our own portfolios to where the CAR-T and gene editing is, we get as close to a cure as possible for these patients to bring the benefit.
So in a way, this brings us back to what Tony said in his address. At the end of your address, you said collaborations are the gold star of pharmaceutical development. And if you have a therapy as complex as mRNA therapeutics across at least a half a dozen different therapy areas, and you have China in the background, you have to approach it from some kind of a new collaboration, I guess. I mean, you're the expert. But I read recently a column by an analyst in the United States about how American companies are looking at Chinese companies from the point of view of using small Chinese companies as a source of new products and new ideas, bringing them back to the US and then testing it out in a context in Europe or in the US. And I'm wondering whether mRNA therapeutics fits into this modality here, more collaboration.
Yep, I think definitely it does. But it's not only mRNA, all modalities fit into that field. I mean, it's for sure that on AstraZeneca and other large farms, we have to, whether the SMEs are based in China or whether they're based in America or Europe, it doesn't really matter. Some Chinese companies seem to progress much faster towards the clinic. So it's a case-by-case basis, not just solely going to focus on China. It's whoever's doing the best science and the best possibility of getting to those patients with a platform that's robust enough to show utility.
Yeah. So when we look at mRNA therapeutics, you've obviously got advantages and you have disadvantages. And one of the advantages that came out of the discussion so far this morning was protein expression. Better, easier, perhaps, than many other therapies. Perhaps you could explain why, and then we can go into the shortcomings, the problems that we still need to address.
Yeah, so maybe I could go for we've got the nice sort of contrast we can have between the lentivectors, the DNA, and the mRNA LNPs, and what sort of the trade-off between those two modalities are, basically trying to achieve the same thing. I think mRNA safety compared to lentivectors. So one of the trials, actually, AstraZeneca, the ESA Biotech one, so they have early data from in vivo lentivector DNA, in vivo CAR-T, like really good efficacy, but safety signals it's a viral thing you're putting in, and that creates a problem. But maybe that's okay if you've got end-stage cancer patients. Where that might not be okay is if you've got an autoimmune disease patient, where that risk-benefit is a different calculation. Mm-hmm. So mRNA is maybe better in that kind of situation, but the reason mRNA is maybe safer is because it doesn't persist as long, which creates its own problems in that you don't maybe have the same level of efficacy. So that's where the balance is going to be, choosing between those two things. And I think it could be that it's neither one of these is going to win, it's just that we're going to use it for different indications, different approaches.
Yeah, so you'd be looking for a different duration of effect depending upon what the disease is.
Yeah, indeed, and a different, say, risk-benefit analysis. On the other modalities, I would mention TCEs, so bispecific antibodies. So there is an argument that these could actually just replace CAR-T. Instead of going in vivo, just put in a TCE. So what a TCE does is it's a bispecific antibody that brings the T cell and the cancer cell together. Engager. So you don't. Yeah, it's just a separate, just like a normal bispecific antibody. Yeah. So then you don't need all this cell reprogramming. You're just like bringing the cells in and activating the cell. So that is another thing that actually could just disrupt the whole in vivo CAR-T space. Actually, maybe it's just TCEs. But then maybe mRNA can help with that in that you could actually express your TCE in vivo as well. So you could put your LNP with an mRNA expressing a TCE, and then that is in the environment. So all of that to come as well.
So if you go back to this question of the disadvantages, or maybe shall we say the weaknesses that still have to be addressed, one of them is duration of effect, and that fits in with the delivery, I suppose. So if you could just discuss duration of effect and delivery. Delivery is quite important because do we know whether the LNP is the ideal delivery vehicle? I don't know.
Yeah, because we've got the targeting problem. So as I mentioned, like your LNP at the moment for the vaccines, not targeted, and they naturally go to the liver.
Yeah.
If you want to target a T cell, you have to work out how you're going to target your LNP. And so you can stick antibodies on, you can stick proteins, you can formulate them in a way that's going to go to T cells more. But that is a challenge. No one has solved that problem in a way that everyone's going to follow yet. So that's another sort of platform development that is going on.
Mm-hmm.
And that's certainly an area that I've seen kind of recently about, companies are developing, the LNPs which have, specific capabilities to try and target particular tissues, and certainly, in terms of patent filings in that delivery area, that's an area which there's been a lot of activity in recently.
I don't know whether this is something you want to discuss, but how do you decide regarding the delivery? Is the LNP well established as the delivery vehicle?
I think the advantage of an LNP is that it's taken up by the endosome pathway. It ionizes, smashes it open, gets into the cytoplasm. So it's a pretty efficient delivery mechanism. Mm-hmm. There are lots of alternative ways of delivering RNA or delivering DNA. But if you deliver DNA, you've got the additional problem of getting into the nucleus for expression. So LNPs have had significant advantages over other technologies and have kind of come to dominate the space. Certainly the IP is complicated. And on the RNA side, the self-amplifying RNAs, they're an attempt to lengthen expression as well as boost it. Circular is an attempt to lengthen expression. Circular RNAs exist in nature. They're somewhat obscure and not generally in undergraduate textbooks, but they are out there. To use those, you need an IRS, which brings in all sort of multi-polycystronic viruses, et cetera, et cetera. So it's a really interesting technology for a biologist. It's got enormous potential. But the lipids, ultimately, if we look back in 50 years, we'll see them as primitive. They're things which have got endless half-lives. No one knows how long they last in the liver. They build up. They don't seem to be toxic, but no one knows. So I think in terms of treatments for common diseases, there's a long way to go, and the technology is not mature. But for effects where, like in hospital infection prevention, cancer vaccines, where you just need to give it a short course, it's absolutely fine and very, very powerful.
Yeah.
So I think one of the advantages of mRNA is that, I think it has been mentioned, so compared to DNA, it doesn't have the propensity to integrate into the genome, whereas DNA has that potential and that risk, that safety risk. mRNA doesn't have that because of the transient nature and also because of the modality. I think, touching on the lipids, you're right. I think we're at the beginning of lipid optimization. So with AI now helping on many different platforms around lipid advancements, or lipid LNP advancements, that's going to really help in the delivery, the specific delivery, whether it's targeted or to do with the functionality of the lipid with regard to the composition, with the duration, and also, I guess one of the down bit plays about LNP delivery of mRNAs is actually the amount that gets released is, dogma is around 1% of what you're delivering actually is effective at releasing as cargo in the cell. So if we can improve on that, there's massive space for improvement in lipids. But I agree with you, there are other delivery vehicles that will, definitely come on the market at some stage in the future, but we don't know what they are yet.
Are you saying that transient expression has a big advantage and also a disadvantage at the same time?
Yes, absolutely. Yeah. So I think it will have its advantages and it will have its places where it can be used because of that advantages. There are other aspects where mRNA may be useful, but because of the transient nature, may not be as applicable. In vivo CAR T, we can give multiple, hopefully with the improvement of lipid immunogenicity. We can give reduction of PEG, perhaps. We can give multiple doses of repeat doses, short, close together repeat doses of mRNA for an in vivo CAR T. So it's generally 2 or 3 doses. So it currently happens for those patients.
So you're talking about in combination or as an adjunct? Or
No, it's the same product, just dosed every 2 to 3 weeks apart.
Okay.
That's how it works for mRNA, as opposed to the vaccines where maybe you get 2 doses once every year or something like that.
Yeah.
So, but with current LNPs, there's the potential issue of ADA because of the PEG.
Yeah. Now, a lot of time was spent so far this morning talking about in vivo, the new future. Who would spend money manufacturing cells when you can let the body do it itself? So is there any follow-on to transient expression and the effectiveness of the mRNA in an in vivo setting? Or does that not matter? I mean...
So I may mention, Cartesian as a company, that is a moment their phase 3 product is a autologous mRNA CAR T.
Yeah.
So it's making the cells in the same way as I showed with the diagram of DNA CAR T,
Yeah.
but with mRNA.
Yeah.
And so that has the disadvantage that those cells don't persist in the same way that DNA CAR T persists when you put them in the body. They disappear. But the huge, massive advantage in that you do not get CRS. Like never has been a CRS with mRNA CAR T. Mm-hmm. And so that means you can actually administer multiple doses, and also you can treat autoimmune diseases instead of just end-stage cancer where the risk benefit is not, where that equation makes more sense. So that is really one of the really good examples of both the disadvantages and the advantages of that transient nature of the mRNA. It goes away, and therefore you don't get...
Yeah. So you might find commercially companies are saying instead of we have one dose that cures you for the rest of your life, you can have multiple doses and get the kind of protection that you need for a disease which is not effectively covered at the moment.
Yeah, and it still has the same efficacy. So so 6 doses remission in autoimmune disease.
I guess it also depends on the condition you're trying to treat. So with traditional gene therapies, there's obviously big disadvantages for the use of viral vectors, but where it's to treat a genetic mutation which is going to persist through the life of a patient, hemophilia or lots of other rare ones, then that sort of single administration with long-term benefit is massively advantageous compared to having to take repeated treatments throughout your lifetime. So I think it depends partly on the condition.
Should we ask if there are questions, please? Because this has been rather technical, and there may be some people out there that would like to ask questions that apply to their jobs and their readings. Yes.
Hello, I have one question looking a little bit into the future, and this is collaboration versus royalty stacking. I mean, if you're looking at the improvement in the field, there are now many companies which specialize in one aspect of it. So you see new patents on, let's say, lipids, then on modification of those lipids, then on formulation of the LNPs. Of course, ideally, you would take the best of all worlds, but at some point, royalty stacking becomes an issue. So is there any ideas on how to make sure that you can actually use the best what you have, and how to handle this? Do you see this actually as a problem for the future, or is this something which just hopefully solves itself?
Yeah, it's a problem. So royalty stacking is a big problem because you've got all these different components. You've got a license in your lipid, you've got a license in your car, you've got a license in what your mRNA is. So I think that is where the platform that does multiple components actually becomes more valuable. But I would say that a lot of the sort of foundational IP within these spaces is expiring, and so there's a huge space now where people can innovate, do their own thing, actually solve a different problem, optimize something in a different way, generate new IP, whilst the older IP is going to actually be expiring. I think that is where the focus can be. So in the ionizable lipid space, I put up the chart of Acuitas with all their ionizable lipids. You don't have to use Acuitas' ionizable lipids because there's hundreds of companies making ionizable lipids. You need to find the ones that actually solve the problem that your particular technology needs to solve. And that is where you actually get your own IP and avoid the having to have all the royalty stacking issues.
Yeah, and I guess just building on that, that's why it's quite important. I guess more important nowadays to think about your freedom to operate in terms of third-party IP from an earlier stage. So as Rose says, there's some of the LNP patents I think are expiring. A lot of them in the next sort of 5 years, and some of the mRNA patents, the early ones, in the next sort of maybe the early and mid 2030s. So it might be that those are less of an issue. So if you can see that there is a bit of a gap once those expire, and you've got an area which is a bit freer to operate in with fewer third-party patents there, then that opens up a bit of a window for you to make your own IP in that space, and also not have to pay the royalties in the same way. So that's a
Yeah, I think you also need to be careful in this field, like not just the royalty stacking in the FDO, but also making sure you're not using someone else's trade secrets. So one of the litigation that's currently happening in LNP in vivo CAR-T space is a trade secret litigation. So Capstan filed patents, and they're now being sued for actually those lipids you filed on were someone else's. Your inventors moved companies, and because the space is so crowded, that is so easy to happen that people move between the companies and share that information. So that also has to be a big consideration as well.
Okay.
I think the main fundamental is always follow the science first, and let the lawyers fight it out later.
Yeah, you can't build IP without the science. The IP is always going to be based on the science, and solving a problem in your industry is what good IP is based on.
Okay. So,
Okay.
yeah, I'm sure we could go on for another hour, but let's let people digest what you've already said.
Thank you, Victoria and panel. Thank you.